mosaicism in human skin.
Our genotype is changing continuously during life, so every one of us is a mosaic. But every kind of lesion on the skin may be explained as a mosaic mechanism. So consequences are really, really unpredictable. But nowadays, everything is changing.
Welcome to this new EADV Podcast, and my name is Antonio Torrelo, and I'm editor-in-chief of the JEACP, and the reason for this podcast is that we have recently published a special issue on mosaicism in human skin. And to comment on this, we have a contributor for this supplement, and also one of the best experts in the world about the field of mosaicism. I'm happy to introduce Professor Andrea Diociaiuti from the Hospital Bambino Gesù in Rome. Welcome, Andrea.
Thank you, Antonio, for this opportunity.
Andrea is as well the leader of the European Reference Network for genodermatosis in the field of cutaneous mosaicism. So, Andrea, happy to have you here. So before we move ahead, can you give us a definition or a concept of what is a mosaic? What is an individual mosaic?
It seems to be an easy question, but I think we are discussing on this definition since the 70s, probably. Mosaicism is a genetical concept, but has also some clinical consequence. So the most classical definition is the definition made in the 70s by Rudolf Appel. It's a mosaic. It's an organism that is composed by at least two populations with different genotype, with different genetic characteristics, but that derives from a single zygote. So from a zygote that is genetically homogeneous. So this is the classical definition, but the problem with this definition is that it's very precise, but it's very comprehensive because every one of us, since life in utero has, and then also after birth, has millions of mutations. So our genotype is changing continuously during life. So every one of us is a mosaic. Definitely. So this is the problem of this definition. So that's why also we made a consensus several years ago in ERN, together with you also, as you remember, when we also talk about mosaic abnormalities on the skin or mosaic disorder, because what we want to study is the mutation, post-zygotic mutation, that cause abnormality on the skin or a disease. So this is the concept.
Very good, very well explained. So, Andrea, with this definition in mind, what are, in your experience, mostly in pediatric dermatology, what are the most common diseases that fall under this classification of mosaicism, so that the listeners can understand what we're talking about?
The range of phenotypes linked to mosaicism is almost infinite, because it depends of course of several factors. But when you have a mutation, a post-zygotic mutation, you can have no change in the amino acid. You can have a change of the amino acid, but it is not mandatory that the structure of the protein will change, but you will have also mutation that will give some difference and some consequence on the phenotype of the and the phenotype depends fundamentally on which is the cell that is the target of the mutation. So you can have mosaicism of keratinocytes, so and epidermal nevi, for example. You can have also connective tissues nevi. You can have pigmentary mosaicism. You can have vascular malformation. I think probably vascular malformation are most of the very, very frequent, because we have a high range of vascular spots in children. So I think that, and also we have to consider also congenital nevi as a mosaicism. So these are, I think, the most important groups of mosaicism.
Absolutely. You have mentioned a couple of words such as nevus, nevi, malformation, and how do you link how do you make an equivalence between these words and mosaic? Is mosaic a better term than referring to these terms such as nevus, hamartoma, malformation? What is your
Yes,
opinion?
yes, the concept of nevus has a genetic concept because there is something of abnormal on the. If we talk of the skin, of course, like a spot or proliferation in a utero of a clonal group of cells that has a genetical origin, and this will lead to a stable lesion on the skin that will not proliferate usually, and this is the concept of nevus. But the problem with this definition that every kind of spot on the skin probably may be considered as a mosaic, as a nevus, and this concept is so wide that is may also change the how we are looking for dermatological disease because. Apart from inflammatory disease or autoinflammatory disease, but every kind of lesion on the skin, there is a single or also multiple lesion and malformations may be explained as a mosaic mechanism. So this is what is very interesting and is changing also the way we are looking for dermatological disease, I think.
And in line with this, I suppose you can recognize mosaicism from a clinical point of view. How can you recognize? How can you be completely aware that a patient has a mosaic condition? Do you do it by clinical grounds, by patterns, by type of lesion? What is your advice?
The most important clue is the clinical observation of the patient. That is really it's really important to observe all the patient. If you can if you observe a segmental pattern, it means that you have a part of the skin that is different from the other part, and usually there are some characteristics. There is asymmetry of this kind of lesion, but there is the midline is respected, and if you see such kind of uh, um, of pattern, you are looking for a mosaicism. But in this segmental pattern, you can distinguish a few different patterns. For example, the narrow band, there is the line that were described from Alfred Blasco in 1901, and he described these lines, but he didn't understand really which is the meaning of this line that then understood in the 70s. Uh, and these are clonal proliferation of cells in the embryo life that are due to the movements of the cells in the embryo, because in the embryo you have two proliferation in the length and in the width, and the speed of this proliferation is not always the same, and so you have drawings of clones of cells like in electrocardiography, for example. So it's a sinusoidal lines that are the Blasco lines that we can observe as patterns of mosaicism. This is one of the most frequent, but you can have also large, broad Blasco lines, and there are more rare. We can see in very rare disease, and you can see also checkerboard drawings in this. These are mostly related to pigmentary mosaicism or vascular malformation, for example, or you can have also phylloid pattern, so like flowers on the skin, very round oval patches. But this is the most enigmatic because we don't know exactly the pathomechanism of this kind of drawings on the skin, and they are related more to trisomy or tetrasomy, for example. You can have also half a body. There is the case of child syndrome, for example. So this is the segmental pattern, but you can have also not segmental pattern. For example, if you have a congenital nevi with a very large patch without midline separation, or if you have one single malformation or multiple lesion. This is the case, for example, of dominant disease, where you can have is a biallelic dominant transmission, there is a mechanism like mostly as of double hit mechanism, when you inherited one mutation that make you predisposed to the disease, but you have others, multiple mutation after post-zygotic mutation that cause the lesion on the skin. This is the case of neurofibromatosis, for example, or sclerosis tuberosa, or capillary malformation and arteriovenous malformation. So these are you can recognize on
Uh,
clinical observation.
Absolutely, yes. And I think you mentioned patterning is the key feature for recognizing an individual mosaic. And in line with this, what are the consequences for a patient who is having a patterned lesion, not only in the skin but beyond the skin? Is there What are the main consequences of being a mosaic?
It depends. You cannot define which is the consequence only observed in the skin. For example, we have a lot of discussion if there is a relation between the distribution on the skin and the involvement of internal organs, but it seems that there is no relation. So you have to make exams to search for other organ involvement. It depends, of course, on the lesion you are observing. So we know that if you have, for example, a vascular patch, probably you can have the involvement of such organs or other kind of complication. For example, you can have coagulopathy, or you can have with the epidermal nevi also rickets, because you can have the literature may help us to know what we have to search based on the lesion we are observing on the skin. But the consequences are really unpredictable, because if we have very variables in monogenic germline disease, the variables are more and more in mosaicism, because it depends on this distribution of the mosaic, and the cells that are really interested by the mutation. So consequences are really unpredictable, but we can have some literature that may guide us, so what to search depending on the lesion on the skin. So this is very useful. And there is also another important question. So if this mutation may survive also in offsprings. So, as a consequence, you may have, it is the case, for example, of epidermolytic nevi, you may have a mutation with a nevus on the skin, but if this mutation is inherited by offspring, they will have a germline mutation. They will develop a complete disease of the skin. There is keratinopathic ichthyosis in this case. So we have these two aspects, and the second aspect must be discussed formally with a geneticist to make appropriate counseling to the family.
So it's clear that mosaicism goes beyond the skin, and it may imply many different consequences according to the complexity of the topic. So recently we have had a lot of advances in genetics, and just briefly, how do our position about mosaics has changed due to the genetic advances in the diagnosis? For example, do we have to diagnose mosaic patients by tissue study, or so? What are the advancements in genetics?
There is a lot of advancement, especially in mosaicism, in genetics, I think, because of the new techniques that are next generation sequencing. The first sequencing of DNA was in 1977 by Sanger, and we historically take biopsy from the skin to have culture of fibroblasts. But the problem with mosaicism is that you can have a mutation not only in fibroblasts but also in other cells, for example, keratinocyte or endothelial cells on vasculature. So the combination of new techniques for extraction of DNA from the whole biopsy, together with next generation sequencing, allow us to have to detect a very small amount of mutated DNA in the biopsy, because with these new techniques we have a very fast response with low expenses, and you can reach also make diagnosis of variant with very very low frequencies. About 1% of variant allele frequency may be detected by next generation sequences, allowing us to make genetic diagnosis of mosaicism in very very difficult cases that in the past was really impossible.
And when a patient receives or the family a diagnosis of mosaicism, I think we can be very precise in determining what is the tissue, the cell involved, what is the extension of mosaicism, the uh, extracutaneous consequences, and we can also disclose perfectly what is the gene involved in mosaicism. With this in mind and understanding the complexity of mosaic mechanisms, how can we treat mosaicism? What can we do for the patients?
Okay. Historically, we in the past we just treat search for complication and treat complication. For example, you may have a mutation in JEACP gene on the face with a capillary malformation, but we will have also seizures. So in the context of Sturge-Weber syndrome, in the past we just treated the complications of mosaicism. Nowadays, everything is changed because we can have the precise mutation that is responsible for the mosaicism, and we can also repurpose drugs that came from oncology. With the concept of precision medicine, you can have a problem, a somatic problem in a part of the body. You can define the mutation, and you will be capable to use a specific drug that hit this mutation in order to treat the patient. Results are discussed. We have a lot of clinical trial in course to study exactly which are the effects of these treatments. We don't know a lot of things. We don't know the long-term side effects of this treatment. We don't know exactly which is the efficacy of this, but we know that there are some nice results. For example, we have some drugs that may hit PK3CA mutations. The classic one was alpelisib, but we have also new drugs that are ongoing on PK3CA. We have also some drugs that may hit mutation in AKT1, for example, for Proteus syndrome, and not only, and also for arteriovenous malformation that usually are in the context of the RAS pathway. We can use some drugs like trametinib that will hit MEK inhibitor, and so will hit the RAS pathway. And so there are a lot of new drugs we are studying, and we hope that in the future will be more feasible to treat really this patient.
So it looks like the field is fascinating, not only from the diagnostic point of view, the very unusual patterns that patients show and the potential consequences and the impact our management and therapies can have on patients' life. So the topic is extremely complex. So how can Andrea, our supplement in the JEACP recently published with you as one of the main contributors, help clinicians to better understand and manage the mosaic patients in the clinical practice.
As you know, we had a scientific day in Rome, and after this scientific day with most of the European experts in this field, you decided to make this supplement. I think it's really a very nice instrument because in this issue of the journal, we approach many practical question in the mosaicism. So the definition, of course, but also the clinical approach to the patient. And so the most frequent mosaicism, so epidermal nevi, pigmentary mosaicism, vascular malformations, and so on, and also the genetic analysis. So I think it's really a practical guide for dermatologists, and not only, also geneticists and also pediatrician, for example, who wants to work in this field, expert or non-expert on this field, and they have a practical guide on how to approach the patient, and with the consensus of experts in this field. So I think it's really a nice job that you made with the contribution of all the experts in the cost and the in context of the European reference network.
So I think it's that the issue is splendid, and I encourage you all to read it, and surely you will get a lot of information in our open access journal JEACP. Thank you very much, Andrea, for your comments. You're such a great expert, and I'm very proud to have you here. And thank you for contributing to the JEACP. Thank you very much for listening.
Thank you, Antonio.